Hi, we are BeneRAS. I'm Roti Johannes, the CEO, and together with me is Rashid Bukai, the CSO. Over 60% of ALL patients are 16 years old or younger. And up to two-thirds of ALL patients experience cancer relapse within 5 years. Survival after relapse is usually less than 5 months. About 30% of ALL patients are caused by mutations in RAS, which is often the root cause of relapse. RAS is notoriously hard to target, highly dependent on the mutation type, and it is considered undruggable. In normal cells, signal of cell replication is received by the EGFR gene, which in turn activates the gene SOS1 and GRP2. These consequently turn on RAS protein, which in turn activates RAF and cascades of gene signaling that leads to normal cell replication. In cancer cell with RAS mutations, the mutant would signal itself on regardless. This leads to uncontrollable replication. Our solution is a liquid-peptide RAS inhibitor that enters the cancer cell, attaches itself to the cell wall, disrupts the mutant RAS, and stops the cancer replication. Our preliminary results show that our drug can target a very wide spectrum of RAS mutations. In this panel, we show 5 cancer cells being tested pairwise against control. We grow the cells for a couple of weeks, and then one of the pairs is treated with a drug at varying concentration. The x-axis shows the drug concentration. The y-axis at 100% means the drug is completely ineffective, and at 0 means that the cancer cells are eliminated. As you see here, that our drug is highly effective. There are about 17,600 patients newly diagnosed with ALL per year in 6 big economies. Of these, 30% are caused by RAS mutation, and on average, each patient spends about $156,000 per year. Multiply these numbers together, we get about $825 million a year. Here is a snapshot of comparison between our drug and the competitors. Our drug poses very low chance of relapse and low toxicity compared to the other standard drugs. Compared to the other RAS inhibitors in clinical trials, our drug can target much wider RAS mutations and thereby more applicable than these drugs. Compared to targeted therapy, stem cell, or immunotherapy, our drug is less expensive. Our primary business strategy is to apply the orphan drug designation or breakthrough therapy designation to reduce time and cost. Then we aim to be acquired by the large pharma company upon successful phase 2. Alternatively, we perform a phase 3 clinical trial with a key partner toward FDA approval in 2032, followed by traditional avenues of drug sales, first in the US, then worldwide. We develop new drugs against other cancers or apply for new indications whenever possible, or sublicense the IP. One of our strategies to prolong our IP is to start a limited warranty program. For example, to assure patients that there's no cancer relapse, we provide a 2-year limited warranty program where, for which in case of a confirmed relapse, we will provide the drug for free and reimburse the medical expenses to a certain amount, say $100,000. Here is the key expenses and milestone, assuming that we can resume preclinical period in 2022. We anticipate our FDA approval at 2032 and 2034, and the break-even point in 2035. 2036 onward, we expect profit greater than $110 million a year. This is not accounting for application of new indications. And assuming a discount rate at 15%, we calculated the NPV to be about $37 million. We have performed some mouse experiments to establish preliminary safety and some pharmacokinetics and pharmacodynamics data. We need to continue PKPD experiments to establish dosing regimen, thorough toxicology tests, thorough safety tests, and to pilot drug production CMC batch. Our goal is to file the IND in 18 months to move forward to clinical trial phase 1. And for that, we need $1.8 million. We also plan to apply for foundational grants to offset the costs. And here is our team. I am Robbie Johannes, the CEO. Present with me is Dr. Shin Mukai, the CSO. We are advised by Dr. Travis Whitfield, and we are partnering with Champions Oncology, represented by Dr. Shabby Chen. Thank you so much.
Thank you so much for that great presentation. Are there any questions?
Hey, Robbie, this is Srinid. I have two questions. One question is that how does your therapy compares with, I see you have shown the competitor slide, but I didn't see the oncolytic viruses covered in that one. So can you comment on the oncolytic viruses versus your therapy? And the second question is that whether this is a direct effect on the KRAS mutation or targeting the downstream inhibitor of the KRAS?
This is the direct targeting direct KRAS mutations. So what was the first question again?
Like how does this compare with the oncolytic viruses, which are, I guess, like the next stage of treatment, and they are more powerful tools as compared to any other inhibitors out there? So,
oh yeah, so basically we believe that our drug is, we haven't done any direct comparisons just yet, but we believe that seeing from the data, we see that it is quite comparable to these oncolytic drugs.
excellent.
Thank you.
Thank you.